Hello there, and welcome into your first lesson for Science Start. This one's going to be all about biological macromolecules and a little bit about polarity and the functional groups that make up macromolecules. My name's Devin, and I'm going to be leading you through this lesson. If you do have any questions about the lesson, I'll be available on the forums or the posting boards, wherever we're discussing, and you can ask me your questions, and I'll be available to answer them there. Okay, so without further ado, mayonnaise is not a macromolecule, but we can start learning about them. So our learning goals for today will be, by the end of the lesson, you should be able to list the various functional groups and know their properties, list the four major biological macromolecules, understand which functional groups make up which macromolecules, and understand how monomers bind to one another to make these larger macromolecules. So we're going to start with a little review about polarity and polar versus nonpolar. So polarity describes the partial charge within a molecule. So it's just the difference of electronegativity between bonded atoms. So all atoms have this measure of electronegativity, which is how well they can attract valence electrons. So that's electrons in their outermost shell. So you can kind of see from the table, the higher values are generally go left to right. As you go left to right, you increase in electronegativity. And as you go bottom to top, you also increase in electronegativity. So fluorine is actually the most electronegative. So like, it kind of makes sense as you go left to right across the periodic table that you increase in electronegativity. Because say you have a full valence electron shell for both nitrogen and fluorine, nitrogen only has 7 protons, so there's a weaker attraction to each electron than fluorine, which has 9 protons, so there's more of a positive charge that will attract the electrons closer to the nucleus of fluorine. And then if you're going up the periodic table, you might think, well, like something like chlorine has 17 protons, so shouldn't it have a stronger pull towards these electrons? That would be true if they're in the same electron ring, but since the valence shell of chlorine is much farther than that of fluorine, it has a weaker attraction to these electrons that are farther away. So fluorine is actually the most electronegative element on the periodic table. So like I said, polarity describes the partial charge within an atom, and electronegativity is it's just kind of a measure of the difference of electronegativity between two atoms, and it's basically just electron sharing. So it's a measure of how well these two atoms share electrons and where the electrons spend most of their time. So fluorine is the most electronegative atom, and then oxygen is pretty electronegative, more than nitrogen, more than carbon, which is more than hydrogen, but they're about equal. So these are the main elements that we're going to be dealing with in our lesson. So nonpolar molecules have the electrons bonded between atoms, and the electrons are shared equally. So as you can see here, although oxygen is very electronegative, it is nonpolar because each oxygen atom has the same amount of attraction towards those shared electrons. But in polar atoms, there's an unequal sharing of these electrons. So for instance, in hydrofluorine, the electrons are more likely to be near the fluorine atom than near the hydrogen atom. So this gives fluorine a partial negative charge and hydrogen a partial positive charge. This creates what's called a dipole within the atom because there are two distinct polar regions. It's also important to note that this is not an ionic bond. It's different from an ionic bond. It's only a partial charge. It's not a full electron being passed from one atom to the other. So that's why these partial charges are denoted with a delta negative or delta positive. Okay, so some quick rules of thumbs for polarity. Symmetrical molecules are always nonpolar. So something like methane, which has a carbon atom with 4 equally spaced hydrogen atoms, will have an equal sharing of electrons between each bond. So it's nonpolar. And just like our O2 example of oxygen, is also nonpolar. Atoms with a lone electron pair are polar. Okay, and then like molecules interact with one another. So nonpolar atoms or nonpolar molecules will interact with nonpolar molecules, whereas polar molecules will interact with polar molecules. And water is polar, and this is a very big molecule that we're going to be discussing because water is found everywhere throughout the human body. So it's in a lot of, like it's in our cells, it's in the intracellular space between cells. So it really determines a lot of the way proteins interact with one another. So if a molecule is polar, it's said to be hydrophilic, meaning that it will interact with water. And if a molecule is nonpolar, it's said to be hydrophobic, which means it will not interact with water. And a good way to just to kind of remember that is like a phobia is a fear of. So nonpolar molecules are almost afraid of water. So they're phobic, so it's hydrophobic. Whereas polar molecules like to interact with it, it's hydrophilic. Okay, so now we can move on to the functional groups. So functional groups is a specific set of atoms bonded in a specific manner that's responsible for the characteristics of an entire molecule. Or if sometimes in very large molecules, the functional group won't determine the function of the entire molecule, but it will determine the function of the local area that it's in. So there are many functional groups that are found throughout biology and biological systems, which include hydroxyl, carboxyl, carbonyls, aminos, and phosphates. And we're going to be discussing all of them here. So the first one we're going to cover is hydroxyl. And hydroxyl is a functional group consisting of a hydrogen atom bonded to an oxygen atom, which is also bonded to any random molecule. So you have your hydrogen bonded to oxygen, and then oxygen can be bound to any organic molecule. So hydroxyl groups are polar and hydrophilic. They're polar because of the unequal sharing of electrons between oxygen and hydrogen. And because they're polar, they interact well with water. So they're hydrophilic. The symbol for a hydroxyl group is OH. And hydroxyl groups indicate that an organic molecule is an alcohol. So whenever you see a hydroxyl group, it means that the organic compound is an alcohol. And then hydroxyl ions, so if it was not bound to an organic molecule, there would just be an extra negative charge here. So they can be bound to many metals and bases. So hydroxyl ions can be a good indicator of how basic a solution is. So if you remember the pH scale, a standard acid that you would use would be hydrochloric acid, which is HCl, and you might neutralize that with a sodium hydroxide base, which is NaOH. So when you put those two together, the hydrogen from hydrochloric acid will interact with the hydroxyl group to make water, and the sodium will react with the chlorine to make salt. Okay, the next group that we're going to be talking about is carboxyl. Carboxyl is a functional group consisting of a carbon atom double bonded to an oxygen atom, which is also bound to a hydroxyl group. So carboxyl groups are polar and hydrophilic, and their symbol is COOH. And carboxyl groups indicate that an organic molecule is a carboxylic acid, as the name would imply. So carboxyl groups are generally acidic, and they will donate the hydrogen atom and gain a negative charge. So they will lose this hydrogen atom or donate it and become negative. And this negative charge can be passed between the two oxygen atoms bound to the carbon. So hydroxyl and carboxyl groups can often react together to create what's called an ester. So in an esterification reaction, the alcohol will react with carboxylic acid to create an ester here, which will link the two organic molecules together and create water. So carbonyls are functional groups consisting of carbon with a double bond to an oxygen atom, which is also bonded to two other organic molecules. If the carbon is bonded to a hydrogen atom, in addition to the double bonded oxygen, it's called an aldehyde, which looks like this. So there's a double bond with, or there's a double bond with oxygen here, and then bound to a hydrogen. But if it's linked to another organic molecule, it's called a ketone. So instead of having a hydrogen here, you have another, it could be a different, could be the same organic molecule. So carbonyl groups are polar and hydrophilic, although depending on the length of the molecule, it may be hydrophobic in some regions. So symbols for an aldehyde are COH, and for a ketone, it's just CO. Now we're moving on to amino groups. So this group consists of a nitrogen atom bound to 2 hydrogen atoms and an organic molecule. It also has a lone pair of electrons freely available. So amino groups are both polar and hydrophilic, and the symbol is NH2. And amino groups are technically under the categories of amines, and they're specifically primary amines. But because we use this primary amine so much in amino acids, it's called an amino group. So amino groups can also accept an additional hydrogen atom with these lone pair of electrons here to become positively charged and becoming NH3. So this property makes amino groups basic. The final groups that we're going to discuss are phosphate groups. And this is a functional group consisting of a phosphorus atom with a double bond with an oxygen atom and single bonded to 2 different hydroxyl groups, as well as an oxygen, which is then bound to an organic molecule. So phosphate groups are polar and hydrophilic, and the symbol is H2PO4. And phosphate group can donate the hydrogen atoms from either of these hydroxyl groups or both. So since they are hydrogen donors, they are considered acids. Okay, so a quick review for these functional groups. We have hydroxyl, and the symbol is OH. It's polar, hydrophilic, and basic. Carboxyl groups, symbol is COOH. They are polar, hydrophilic, and acidic. Carbonyls can either be an aldehyde or a ketone. They're polar, hydrophilic, and they're weakly basic, more neutral than basic. Amino groups, symbol is NH2. They're polar, hydrophilic, and basic. And phosphates have the symbol H2PO4. They're polar, hydrophilic, and acidic. Okay, so that was kind of a lot of information condensed there, just so that we can get to the main part of the lesson. So if you want, you can take a break here. If you want, you can listen to the Take a Break song from the Hamilton soundtrack, or take a break and watch the entire musical on Disney Plus if you have that. I would definitely recommend doing that. So you can pause the video here and take a break if you would like, or we can keep going. So biological macromolecules. Biological macromolecules are very large organic molecules that perform various different biological functions within the cell. All macromolecules are made up of a unique monomer that produce polymers. So just a string or series of these monomer units. And there are 4 main classes of biological macromolecules. They are carbohydrates, lipids, proteins, and nucleic acids. Okay, so we'll start with carbohydrates. So carbohydrates are more commonly known as sugars, and these are macromolecules that provide cells with short-term energy and can provide some structural support. So the general formula for carbohydrates is CH2ON, where N is the number of carbons in the carbohydrate. So if you think about, say, glucose, that one is C6H12O6. Carbohydrates are classified as either monosaccharides, disaccharides, or polysaccharides. And we'll get into what those are in a minute. So monosaccharides are the monomeric unit for carbohydrates. So that's the base unit for carbohydrates. So monosaccharides. So these are the simplest sugars, and they're mono meaning one, so one sugar. So monosaccharides are made up of 3 to 6 carbons. There are some that can have 7, but in general, they're 3 to 6. The class of monosaccharides is determined by the number of carbons followed by ose. So those with 5 carbons are called pentoses, and those with 6 are hexoses. They're also defined by the presence of either an aldehyde, which would be an aldose, or a ketone, which would be a ketose. The carbons in a monosaccharide are always numbered, and they're always numbered starting at the aldehyde. So this would be carbon 1, carbon 2, carbon 3. Then for glucose, this would be carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6. So monosaccharides can be either linear or in the shape of a ring. So most of the larger ones take the shape of a ring, where the aldehyde will react with the hydroxyl group on the 5th carbon to create this ring where the hydrogen from the oxygen is passed on to this oxygen, creating the hydroxyl group there, and this O shares a bond with carbon 1 and carbon 5. Okay, so there are a lot of very common monosaccharides, and some of these are glucose, fructose, and galactose. So you probably hear a lot about these. Like glucose is used in cellular respiration, and it's broken down to create ATP, which we use for energy. Although other sugars can be broken down into ATP as well, glucose is just the general one that we use when you're first learning about cellular respiration. Another common monosaccharide is ribose, which we will discuss later when we're talking about nucleic acids. And then one other thing to note is that glucose, fructose, and galactose are all structural isomers, meaning that they have the same chemical formula, C6H12O6, but they have different structures. So you can see each of these corners here represents a carbon atom. So you have 1, 2, 3, 4, 5, 6 carbons. 1, 2, 3, 4, 5, 6 carbons. And then you would also have 6 oxygen atoms and 12 hydrogen atoms. So isomers are very common in carbohydrates. Because monosaccharides can also be stereoisomers, such as glucose. So D-glucose has the hydroxyl group on the right side of carbon 5, this one here, whereas L-glucose has it on the left side. That's where the L comes from. So D-glucose is synthesized naturally by plants and is used all throughout the environment, whereas L-glucose can only be artificially synthesized in a laboratory setting, and it's not found naturally throughout nature. And then there could also be stereoisomers in the ring formation of these monosaccharides. So alpha glucose has the hydroxyl group on carbon 1 below the plane of the molecule here, whereas beta glucose has it above the plane of the molecule here. Humans are able to metabolize alpha glucose because of the bond here. We have enzymes that can recognize it when it's bound to another monosaccharide through these alpha bonds, but we are unable to synthesize beta glucose because the bonds are above the sugar. So we still have enzymes capable of metabolizing these and breaking down these bonds, whereas plants do. So it's just very interesting. So like some isomers are not found in nature at all, and some we as humans cannot use at all. So moving on to disaccharides. Disaccharides are formed between 2 monosaccharides. So 2 meaning di, or sorry, di meaning 2. And these monosaccharides are formed through a dehydration or condensation reaction. So you have the hydroxyl group from carbon 1 with the hydroxyl group from carbon 4 of glucose here. These will bind together to create a glycosidic linkage and create a water. So you are dehydrating these sugars, which is why, so you're removing the water, which is a dehydration. So that's why it's called dehydration, or it's a condensation reaction because you're bringing two together and condensing the molecule together. Yeah, so this bond is called a glycosidic bond or glycosidic linkage, and they can be made from alpha or beta hydroxyls. But like I said, humans can only metabolize these alpha ones. If it was above, we are unable to metabolize those. We don't have any enzymes capable of that. So some common disaccharides are sucrose, which is made from glucose and fructose, lactose, which is found in like milk products. So sucrose is like your basic table sugar. Lactose is found in milk. Like you know that people are like lactose intolerant. They are unable to metabolize this. And this is from galactose and glucose. And maltose, which is found in like malt vinegar, is two glucose molecules bound together. Okay, polysaccharides are long chains of monosaccharides. So polysaccharides can be used as energy storage or to help with some structure and support of the cell. So polysaccharides can be unbranched, meaning these glycosidic linkages are only through carbon 1 and carbon 4 of each molecule. So that'll be something like amylose. So they're very linear and straight, or they could be branched, which means that they have these carbon 1 and carbon 4 linkages, but then they are also linked by carbon 1 and carbon 6 here, which creates branching of the macromolecule. So something like amylopectin is made up of only glucose, like amylose is, but the branching causes it to have a different function than something like amylose. So some common polysaccharides are amylose and amylopectin, as we saw in the last slide. Glycogen, which is a bunch of highly branched glucose molecules. So they have a lot of these carbon 1 and carbon 6 bonds. And you probably heard of glycogen because of like diabetic people. So when you eat a lot of sugar, insulin will tell your body to absorb these glucose molecules and form glycogen so that your body has them later for when you undergo like physical activity or something like that. They can be broken down by glucagon and easily create energy that your body requires. So diabetics can lack insulin, which is why they need to take additional insulin shots in order to help them form these glycogen molecules. Starch is another form of branched glucose molecules, but it's not as highly branched as glycogen. And then there's also cellulose, which is made up of beta bonds between beta glucose molecules. So a cool thing about cellulose is that each glucose molecule is actually upside down from the one beside it. So you can see here, carbon 6 is above, and here carbon 6 is below, above, below. This goes on and on and on, which makes cellulose very dense, which makes it very strong. So plants will use cellulose in their cell walls in order to protect themselves because they can fit together very nicely. And then also, because these are made up of exclusively beta glucose, humans cannot digest cellulose. So if you think about plants such as like corn, for instance, the shell of corns are very dense with cellulose. So our body is unable to digest it. And that's why after you eat corn, you may see it once you relieve yourself because your body was unable to digest the shell of the corn, but was able to digest all the interior of the corn. So yeah, that's why you can see the corn in your poop. Okay, moving on to lipids. So lipids or fats are biological macromolecules that provide cells with long-term energy storage and make up biological membranes. So lipids are hydrophobic, meaning that they do not interact well or at all with water. So lipids are made up of, in general, are made up of a glycerol molecule and fatty acids. So glycerol is an alcohol consisting of 3 carbon ions bound to 3 hydroxyl groups. 1, 2, 3, 1, 2, 3. And then fatty acids are long chains of hydrocarbons, which are molecules of just carbon and hydrogen that end in a carboxylic acid. So these chains are generally 12 to 8 carbons long, but they can go as long as 36 carbons, or be as short as, I believe, 4. And then they can be saturated or unsaturated, which we will get into in just a moment. So the carboxylic acid from fatty acids will react with the hydroxyl group to perform an ester bond. So that comes back to the esterification process that we were talking about earlier. So these 3 fatty acid chains react with each carboxylic acid till each carbon in the glycerol has an ester bond with a fatty acid here, and it also produces 3 water molecules. So once the glycerol molecule has been bound to 3 fatty acids, it's called a triglyceride. And then these fatty acids don't have to be the same length at all. They can all be different, and they can be saturated or unsaturated. Saturated fatty acids are fatty acids where every carbon atom has a single bond to each other. So they are completely saturated with hydrogen atoms. In unsaturated fatty acids, they have 2 or more carbons that share a double bond with one another. So here you can see that these 2 carbons share a double bond, so they're not saturated with hydrogen, hence the term unsaturated. And then you can either have cis unsaturated fatty acids where the chain continues on the same side, or you can have trans unsaturated fatty acids where the chain starts on either the bottom here, then is on the top of this atom, or vice versa. It would be the bottom here and the top here. If there's a single double bond in the fatty acid, then it's what's known as monounsaturated, and if there's more than one double bond, it's known as polyunsaturated. So saturated fatty acids are generally solid at room temperature because the fatty acids kind of line up nicely together and can stack easily on one another, which will make it more stable. Whereas the unsaturated fatty acids have these kinks, which don't make them line up as easily, which means they move around more, which makes them a liquid at room temperature. So if you ever, like, you might hear a lot in like the media about like trans fats, and like in something like butter or margarines or something like that, and generally these trans fats are artificially produced from cis fatty acids. So they'll be artificially made from a cis fatty acid into a trans fatty acid, and that's more of why they're not as good for you. Okay, so phospholipids. You might hear this a lot when it comes to cell membranes because they are the biggest component of cell membranes and other intracellular membranes. So phospholipids have both hydrophilic and hydrophobic portions. And this is known as, so it's hard for me to pronounce, amphipathic. You won't hear me say that again throughout this presentation. Okay, so phospholipids are made up of a glycerol molecule, 2 fatty acids, and a modified phosphate group. So here's your glycerol molecule here. You have 2 fatty acid chains, which are known as the nonpolar tails of the fatty acid. And then you have the modified phosphate group here, which is hydrophilic. So this whole top region is known as the polar head. So you can also hear these be termed as hydrophilic head and hydrophobic tails, in addition to nonpolar and polar. So these So the phosphate heads will always face the outside of the phospholipid bilayer because it's hydrophilic, and the environment it's in is usually aqueous or water-based. So this will interact with the outside environment, which is primarily polar. The hydrophobic fatty acids will face away from the outside environment, and they will face each other. So that's why the tails are always kind of in the inside, and the hydrophilic heads are always on the outside of the membrane. And then generally, these fatty acids have at least one unsaturated fatty acid. So this prevents the phospholipids from becoming like packed together, like we talked about earlier. Saturated fats can pack together easily because they can line up nice. But you want the membrane to be fluid to allow molecules to pass through it, and also allow molecules, membrane-bound molecules, to stay within the membrane. So unsaturated fatty acids just prevent the fatty acids from packing together and allow fluidity throughout the membrane. Okay, so steroids. Steroids are lipids that have 4 fused carbon rings together and may have a tail off of one of these rings. So here are the 4 fused carbon rings, and sometimes they can have a tail. So common steroids include cholesterol, cortisol, testosterone, and estrogen, among many others. And although they do not contain fatty acids or glycerol, they're hydrophobic like many lipids, which is why they are categorized under the lipid umbrella. Okay, proteins. So proteins are biological macromolecules that provide cellular structures and chemical signals, increase the rate of chemical reactions, respond to stimuli, transport materials throughout and across within the cell. Basically, proteins do everything throughout the cell. Short of provide the cell with energy and pass on genetic information, proteins do everything else. And amino acids are the monomer for proteins. I might be a little biased because a lot of my research was based around proteins, but I think proteins are the most important one. Okay, so amino acids. So amino acids consist of a central carbon atom, also known as the alpha carbon, bonded to an amino group, a carboxylic acid group, a hydrogen atom, and then a side chain. So here is the basic structure. So you have your alpha carbon here, bound to an amino group and a carboxyl group, and this side chain. And the side chain are different, but they are specific to the specific amino acid. So amino acids bond with one another through the amino group of one amino acid and the carboxylic acid of another amino acid to produce a peptide bond. So one of the hydrogen atoms here will react with the hydroxyl group of the carboxyl group, which will form a water and create what's known as a peptide bond between the nitrogen and carbon. So a series of amino acids all bonded together forms what's called a polypeptide chain. So there are 20 amino acids that make up proteins within humans, and they're categorized based on the properties of their side chain. So here you can see this would be the alpha carbon here bound to carboxyl and amino. So all of these parts are the same for all these molecules, but then it's the side chain here that differs, and they are categorized together based on the properties of this side chain. So that can be a positively charged side chain, a negatively charged side chain, an uncharged side chain, but the side chain is polar, an uncharged and non-polar side chain, or hydrophobic, and these ones would be hydrophilic. And then there are a couple that are special cases which don't really fit in with any of the other categories. So basic protein structure. So the shape and structure determine the function of a protein. So however it's kind of formed, and whether it's like globular or more fibrous, can determine what the function of that protein is. So there are 4 levels to classify the structure of proteins. That's the primary structure, the secondary structure, the tertiary structure, and the quaternary structure. So we'll start with the primary structure, which is just the polypeptide chain, which is the order and sequence of amino acids that make up the protein. So here you have the start. So proteins are always kind of red from N tournaments to C tournaments. So that's how they're kind of made. So they will always start at the N tournaments, which is the side with a free amino group, and they will end at the free carboxyl group here. So this one started with, and then go through with synthesized all the way here to the end. So that's just the primary structure here. Just the sequence of the amino acids that make up the protein. Secondary structures are shapes that begin to form from the polypeptide chain kind of interacting with itself at a very localized level. So this would be in between the amino groups themselves, not the side chains. So it's generally hydrogen bonds that form between the amino acids, and in the majority of cases, it's not the entire polypeptide chain, it's just a small section of it. So there are two different types of secondary structures. Alpha helixes, which are kind of like these corkscrew-shaped structures and kind of go towards more globular proteins, and beta pleated sheets, which are these big kind of more rigid sheets of sections, and these correspond more to more fibrous proteins. Okay, tertiary structures are kind of like the overall 3D structure of the protein, and these structures are caused by the R groups from the amino acids interacting with each other. So with secondary structures, it was more of just like the amino groups and the carboxyl groups of the amino acids kind of interacting with each other. But here is more of the side chains that interact with one another. So some of these interactions include disulfide bridges, which are bonds between two sulfur atoms between these side chains. Hydrogen bonds can form between different side chains. Ionic bonds, so you could have a positively charged and a negatively charged side chain interacting with each other. Hydrophobic interactions, so two hydrophobic side chains interacting with each other, or hydrophilic interactions, so two hydrophilic side chains interacting with each other. So this kind of gives like the bigger shape of the protein. And then quaternary structures. So some proteins are made up of more than one polypeptide chain. The polypeptide chain is limited to the tertiary shape, but then if protein does need more than one polypeptide chain to actually function, then the quaternary structure shows how these subunits interact together to form a complete protein. So for instance, in hemoglobin, the protein in red blood cells that carry oxygen within the red blood cell are actually made up of 4 subunits. So like 2 alphas here and 2 beta subunits. And then it's important to note that not all proteins have quaternary structures. So if a protein only has a single subunit and doesn't need more than one subunit in order to function, it won't have a quaternary structure. It'll only have a tertiary structure. So the quaternary structure is just how the different polypeptide chains interact with each other to form a completed protein. So just a little review there. So protein structures are made up of the primary structure, which is just the polypeptide sequence, and then the secondary structure, which can be alpha helices or beta pleated sheets, and then the tertiary structure, which is like the whole 3D shape and like the side chains interacting with each other, and then the quaternary structure, which is different polypeptide chains interacting with each other to create a completed protein. So the different levels of protein structure can yield one of 3 kind of proteins. You can have fibrous proteins, which are more rigid. I believe I was talking about this earlier, but here are the formal definitions. So these are rigid proteins that kind of help provide cells with structure and support. Globular proteins that are more fluid and move freely throughout and between cells. So these can be more of your signaling kind of molecules. And membrane proteins, which are bound either throughout the cell, throughout the membrane, sorry, or just kind of surfacely to the membrane and help transport materials and receive signals. And then some other kind of types of proteins. You can have enzymes, which are biological catalysts that increase the rate of reactions by binding to specific substrates. So these enzymes are very specific for their specific reaction. Hormones, so these are biological signals that allow cells to communicate with one another and trigger biological reactions. Like when you're going through puberty, you have hormones flying all throughout your body. Transport proteins, which move materials across the cell membrane or within the cell. Receptor proteins, so these proteins are usually on the cellular membrane and they receive signals to trigger a response somewhere in the cell. And structural proteins, so these are the fibrous ones which just help with the cell shape and support the shell from kind of collapsing in on itself. And our final biological macromolecule that we're going to be talking about, nucleic acids. So these are the macromolecules that store and pass on genetic information of the cell. So nucleotides are the monomer for nucleic acids. And there are two main types of nucleic acids. We have deoxyribonucleic acid or DNA, and ribonucleic acid, also known as RNA. So nucleotides. All nucleotides are made up of three components. You have the nitrogen base, the pentose sugar, and the phosphate group. So starting with the nitrogen bases, so these are organic molecules that are nitrogen rich, and there are two different classes of nitrogen bases. You have your purines and your pyrimidines. So purines have two nitrogen rich rings, here and here. So the two different purines are adenine and guanine, also known as just A or G in shorthand. And then the pyrimidines have one nitrogen rich ring, and they are cytosine, thymine, and uracil, or C, T, and U. And then thymine is only found in DNA, whereas RNA only has uracil. And they kind of replace each other in DNA and RNA. So purines will always bind with pyrimidines so that you have three rings in the bond. So you always have 1, 2, 3 rings, 1, 2, 3 rings. So adenine binds with thymine in DNA or uracil in RNA, and guanine always binds with cytosine. So these bonds between the nitrogen bases are hydrogen bonds, and adenine and thymine or uracil have 2 hydrogen bonds, whereas guanine and cytosine have 3 hydrogen bonds. So the bond between guanine and cytosine is a little bit stronger than those found between adenine and thymine. The pentose sugar. So ribose is a pentose sugar found in RNA, and deoxyribose is a pentose sugar found in DNA. So in ribose, on the second carbon here, you have a hydroxyl group, whereas in deoxyribose, on the second carbon, you only have a hydrogen atom. So in other words, this deoxyribose is deoxygenated. It's a deoxygenated ribose, right? Right, get it? Scientists, we are very creative with our naming strategies. Except for the ones from like back in the day, like the ones who came up with developmental pathways, like the sonic hedgehog pathway. That's an actual pathway in biology. You can look it up after this lecture. Or you can pause the lecture now and go look it up now. Okay, back to the task at hand. Phosphate groups. A phosphate group is bound at the carbon 5 of the pentose sugar. Phosphate group will react with the hydroxyl group on carbon 3 of another pentose sugar, creating what's called a 5' 3' phosphodiester linkage. So instead of saying like carbon 5 and carbon 3, we just call them like 5' and 3'. So this phosphate here is a little modified. So the phosphate from carbon 5, 1, 2, 3, 4, 5. This hydroxyl group will react with the hydroxyl group on carbon 1, 2, 3, and create a phosphodiester linkage here between the two ribose, or between the two nucleotides. So these phosphodiester linkages kind of chain together all the different nucleotides and form what's called the phosphate backbone of the nucleic acid. So the structure of DNA, you may know this from the discovery by Watson and Crick, which was stolen from Rosalind Franklin, but that's a whole different lecture. So DNA is formed by two complementary strands. The phosphate backbone on each strand is found on the outside with nitrogen bases found within to allow for binding. So these two strands form what's called a double helix structure. So you can see there's one helix strand here and then another one here. So you have a minor groove and a major groove. The major groove is just bigger than the minor groove. So you can see the base pairs are linking up together in the middle here. And this blue here is the phosphate backbone. So the two strands in DNA always run anti-parallel, meaning that they run in opposite directions, which allows for optimal binding. So you can see the 5' up here, which is from the carbon 5, starts up at the top and goes all the way down to the 3' end. Meanwhile, this one starts at 5' down here and goes up to the 3' end here. So they're parallel to each other so that they bind well, but they're anti to allow for better binding between the two. And it also helps with DNA replication, which we will get to later in another lecture. In the nucleus, DNA is often condensed into chromosomes. So you have these big long strands of DNA that get very packed together using histones and other kind of proteins, but we won't get into that there. That'll be more for your molecular genetics. Okay, so DNA versus RNA, what's the difference? So DNA is double-stranded, whereas RNA is single-stranded. So as you can see here in Daenerys' hair, there are two strands here, like DNA, whereas there's only one strand here, like RNA. DNA uses the deoxyribose sugar, whereas RNA uses the ribose sugar, or an oxygenated deoxyribose, but just call it ribose. DNA uses the thiamine nitrogen base, whereas RNA uses uracil. So DNA is hereditary, whereas RNA is not hereditary. So there's a long time where people weren't sure if it was DNA or RNA that was that was a hereditary material. They also thought it could have been proteins, but whatever. But yeah, so DNA was passed on from cell to cell. So DNA is transcribed into RNA, whereas RNA is translated into proteins, which is like the central dogma of molecular genetics. But again, that'll be in your molecular genetics lesson. DNA is found only in the nucleus, whereas RNA is transcribed in the nucleus, but then found primarily in the cytoplasm of the cell. Okay, so a review of the 4 biological macromolecules. We have carbohydrates, which are made up of monosaccharides and provide short-term energy storage and some structural support. Lipids, which are made up of glycerol and fatty acids. Not exclusively though, because we do have steroids and phospholipids, which are a little bit different. But they provide long-term energy storage and can make up biological membranes. Proteins, which are made up of amino acids and do just about everything in the cell that isn't related to the other biological macromolecules. And nucleic acids, which are made up of nucleotides and store and pass on genetic information. Okay, so you've now reached the end of the lesson. Congratulations if you're still listening. And like I said, I will be on the forum or boards or whatever we're using to communicate throughout these lessons to answer any questions you have. And I'm also open to any feedback. So any ways that we can improve this lesson. I try to throw in some little memes here and there just to kind of keep it interesting for you. But yeah, there is a quiz associated with this lesson that you can do. And I hope you enjoyed your first lesson in the Science Start course. And I hope you enjoy the rest of the Science Start course. Okay. Have a great day.